Hi, in this presentation, we are going to learn about secretory functions of the alimentary tract. Topics that we are going to cover are general principles of alimentary tract secretion, secretion of saliva, esophageal secretion, gastric secretion, pancreatic secretion, secretion of bile by the liver, functions of the biliary tree, secretions of the small intestine, and finally, secretions of the mucus by the large intestine. Here you see a typical function of a glandular cell for formation and secretion of enzymes and other secretory substances. Here you see the nerve, probably autonomic nervous system, uh, it comes from. So it stimulates or it inhibits the function of this gland. In this secretory cell, what we see, this is the nucleus here, and this is the granular endoplasmic reticulum. You see the ribosomes around, and when secretion started to be synthesized here, probably lipoproteins, they will be transferred to the Golgi apparatus, and then after final modification in the Golgi apparatus, they will be stored in vesicles, and then when they are ready, they will be excreted into extracellular fluid or into a secretory glands canal. Okay, if it is now we are talking about external secretion, I mean exocrine function, not endocrine. So these secretions will go to the lumen of the gland canal. Okay. Here you see the formation and secretion of saliva by a submandibular salivary gland. So the secretion starts in the gland. It contains epithelium, alpha amylase enzyme. This enzyme digests carbohydrates, and then it has mucus and extracellular fluid. When secretion travels to the channel, as you see, sodium is going to be absorbed with chloride, and then whereas potassium and bicarbonate are secreted into the channel. So the secretory glands serve two primary functions in the alimentary tract. First, digestive enzymes are secreted in most areas, and the second, mucus glands provide mucus for lubrication and protection of all parts of the alimentary tract. The purpose of this lecture is to describe the alimentary secretions and their functions as well as the regulation of their production. General principles of alimentary tract secretion. Contact of food with the epithelium stimulates the secretion. Direct mechanical stimulation of glandular cells by food causes the local glands to secrete digestive juices. In addition, epithelial stimulation activates the enteric nervous system of the gut wall. The stimuli that accomplish this are tactile stimulation, chemical irritation, and gut wall distension. Parasympathetic stimulation increases the rate of glandular secretion. This is true of salivary glands, esophageal glands, gastric glands, the pancreas, Brunner's glands in the duodenum, and the glands in the distal portion of the large intestine. Secretion in the remainder of the small intestine and in the first two-thirds of the large intestine occurs mainly in response to local neural and hormonal stimuli. Sympathetic stimulation can have a dual effect on glandular secretion. Sympathetic stimulation may increase or decrease glandular secretion depending on the existing secretory activity of the gland. This dual effect can be explained as follows. Sympathetic stimulation alone usually slightly increases secretion. If secretion has already increased, superimposed sympathetic stimulation usually reduces the secretion because it reduces blood flow to the gland. Secretion of saliva. Saliva contains a serous secretion and a mucous secretion. Serous secretion contains betialin, also called alpha amylase, which is an enzyme for digesting starches, carbohydrates. The mucous secretion contains mucin for replication and for surface protection, so it makes swallowing uh, easier. Saliva contains high concentrations of potassium and bicarbonate ions and low concentrations of sodium and chloride ions. Salivary secretion is a two-stage operation. The primary secretion from the acinine contains ptyalin and/or mucin in a solution with an ionic composition similar to that of extracellular fluid. The primary secretion is then modified in the ducts as follows. Sodium ions are actively reabsorbed and potassium ions are actively secreted into the ducts. An excess of sodium reabsorption creates a negative charge in the salivary ducts, causing chloride ions to be reabsorbed passively. Bicarbonate ions are secreted into the ducts, caused in part by exchange of bicarbonate for chloride ions, but also by an active secretory process. Here you see daily secretion of intestinal juices. Saliva has a pH between 6 to 7, and about 1 liter of saliva is secreted in a day. Gastric secretion pH varies between 1 to 3.5 and is secreted about 1.5 liters per day. Pancreatic secretion, again, 1 liter per day, and the pH is a little bit alkalic, varies between 8 to 8.3, and bile is also a little bit alkalic, and then around 1 liter in a day. Small intestinal secretion is also alkalic. pH varies between 7.5 to 8 and about 2 liters per day. Brunner's gland secretion and large intestinal secretion, they are about 200 milliliters per day, and then they are also alkalic. So in a day, our gastrointestinal system secretes about 7 liters of secretions to the intestinal lumen in a day. Salivation is controlled mainly by parasympathetic nervous signals. The salivary nuclei in the brainstem are excited by taste and tactile stimuli from the tongue, mouth, and pharynx. Salivation can also be affected by higher centers of the brain. For example, salivation increases when a person smells favorite foods. Here you see the parasympathetic nervous regulation of salivary secretion. We have submandibular, sublingual, and parotid salivary glands and the tongue. So we have tractus solitarius here, and nervous connections. You can see superior and inferior salivary nuclei send signals to submandibular gland via facial nerve, and as you see here, we have glossopharyngeal nerve innervating parotid and parotid gland and the tongue. So taste and tactile stimuli stimulates the secretion of these salivary secretions, serous and mucous secretions. Serous secretion mainly from parotid gland. We also see the otic uh, ganglion here and submandibular ganglion here. Now, gastric secretion. The stomach mucosa has two important types of tubular gland. Uh, First, the oxyntic acid-forming glands, and the second, the pyloric glands. The oxyntic or acid-forming glands are located in the body and fundus of the stomach. They contain three types of cells. Mucous neck cells. They secrete mainly mucus, but also some pepsinogen, and peptic cells, which secrete pepsinogen. Peptic cells are also called as chief cells, and parietal cells. Parietal or oxyntic cells, which secrete hydrochloric acid and intrinsic factor. Intrinsic factor is important for the absorption of vitamin B12, which is needed for blood formation. And pyloric glands, which are located in the antrum, secrete mainly mucus for protection of the pyloric mucosa, but also some pepsinogen and importantly the hormone gastrin. You see here oxyntic gland from the body of the stomach. So this is the mucosa of the stomach, gastric pit, and then you see the mucous neck cells here releasing, secreting mucus, protecting the mucosa, and oxyntic parietal cells secreting hydrochloric acid, and then peptic cells, pepsinogen is secreted from them. So enterochromaffin cells are also shown here. Histamine stimulates the hydrochloric acid via these cells. Gastric acid is secreted by parietal cells. When these cells secrete their acidic juice, the membranes of the canaliculi empty their secretion directly into the lumen of the oxyntic gland. The final secretion, entering the canaliculus, contains concentrated hydrochloric acid, potassium chloride, and small amounts of sodium chloride. So you see here an oxyntic cells. So the secretion is first into the uh, canal inside and then to the lumen. Here you see postulated mechanism for secretion of hydrochloric acid. Uh, you see here uh, that carbon dioxide comes from extracellular fluid and it is combined with the hydroxyl ion forming bicarbonate and then exchanged by chloride ion. In the luminal side, you can see hydrogen potassium ATP is pumped, pumping hydrogen into the lumen. And what else? You can see that the chloride is diffusing from the extracellular fluid when a transcellular way to the lumen of the plant. Water diffuses by osmosis. You know, the diffusion of water is called osmosis. Hydrochloric acid is as necessary as pepsin for protein digestion in the stomach. The pepsinogens have no digestive activity when they are first secreted. However, as soon as they come into contact with hydrochloric acid, and especially when they come into contact with previously formed pepsin plus the hydrochloric acid, they are changed to form active pepsin. Parietal cells also secrete intrinsic factor. Intrinsic factor is essential for absorption of vitamin B12 in the ileum. When the acid-producing cells of the stomach are destroyed, which often occurs with chronic gastritis, the person develops not only achlorhydria but often also pernicious anemia owing to failure of the red blood cells to mature. Basic factors that stimulate gastric secretion are acetylcholine, gastrin, and histamine. Acetylcholine excites secretion of pepsinogen by peptic cells, hydrochloric acid by parietal cells, and mucus by mucus cells. In comparison, both gastrin and histamine strongly stimulate secretion of acid by parietal cells but have little effect on the other cells. Acid secretion is stimulated by gastrin. Nerve signals from the vagus nerves and local enteric reflexes cause gastrin cells or the G cells in the antral mucosa to secrete gastrin. Gastrin is carried by blood to the oxyntic glands, where it strongly stimulates parietal cells and peptic cells to a lesser extent. Histamine stimulates acid secretion by parietal cells. Whenever acetylcholine and gastrin stimulate parietal cells at the same time, histamine can enhance acid secretion. Thus, histamine is a cofactor for stimulating acid secretion. Pepsinogen secretion is stimulated by acetylcholine and gastric acid. Acetylcholine is released from vagus nerves or other enteric nerves. Gastric acid probably does not stimulate peptic cells directly but elicits additional enteric reflexes. When the ability to secrete normal amounts of acid is lost, the pepsinogen level is low even though the peptic cells are normal. Gastric secretion is inhibited by excess acid in the stomach. When the pH of gastric juice falls below 3, gastrin secretion is decreased for two reasons. First, the high acidity stimulates the release of somatostatin from the delta cells, which in turn depresses gastrin secretion by the G cells. And the second, the acid causes an inhibitory nervous reflex that inhibits gastric secretion. This mechanism protects the stomach. There are three phases of gastric secretion: cephalic, gastric, and intestinal. Here you see the phases of gastric secretion and their regulation. When we see the food, cephalic phase starts. When we smell uh, delicious food, then the cephalic phase starts via nervus vagus. uh, Secretions of the digestive system. And then when the food arrives, and we eat the food, it arrives to the stomach, then gastric phase starts. And then when the food is digested, and then the chyme starts to arrive to the small intestine, and then the intestinal phase starts. So in the cephalic phase, parasympathetics excite pepsin and acid production. In gastric phase, local nervous secretory reflexes, vagal reflexes, and gastrohistamine stimulation all increase gastric secretions, hydrochloric acid, pepsinogen, etc. And then intestinal phase, nervous mechanisms and the hormones released from the intestine affects the secretion in the stomach. So, there are three phases of gastric secretion. First, the cephalic phase. The cephalic phase accounts for 30% of the response to a meal and is initiated by anticipation of eating and the odor and taste of the food. It is mediated entirely by the nervus vagus. The gastric phase. Gastric phase accounts for 60% of the acid response to a meal. It is initiated by distension of the stomach, which leads to nervous stimulation of gastric secretion. In addition, partial digestion products of proteins in the stomach cause gastrin to be released from the antral mucosa. The gastrin then causes secretion of a highly acidic gastric juice. The intestinal phase. The intestinal phase is responsible for 10% of the gastric secretion. It is initiated by nervous stimuli associated with distension of the small intestine. The presence of digestion products of proteins in the small intestine can also stimulate gastric secretion via a humoral mechanism. Chyme in the small intestine inhibits secretion during the gastric phase. This inhibition results from at least two influences. First, enterogastric reflex. The presence of food in the small intestine initiates this reflex, which is transmitted through the enteric nervous system and through the extrinsic parasympathetic and vagus nerves. It inhibits stomach secretion. The reflex can be initiated by distension of the small bowel, the presence of acid in the upper intestine, the presence of protein breakdown products, or irritation of the mucosa. Hormones. The presence of chyme in the upper small intestine causes the release of several intestinal hormones. Secretin and gastric inhibitory peptide are especially important for inhibition of gastric secretion. Pancreatic secretion. Digestive enzymes are secreted by the pancreatic acini. More important enzymes for digestion of proteins are trypsin, chymotrypsin, and carboxypolypeptidase, which are secreted in the inactive forms of trypsinogen, chymotrypsinogen, and procarboxypolypeptidase. The pancreatic digestive enzyme for carbohydrates is pancreatic amylase, which hydrolyzes starches, glycogen, and most other carbohydrates except cellulose to form disaccharides and a few trisaccharides. The main enzyme for fat digestion is pancreatic lipase, which hydrolyzes triglycerides into fatty acids and monoglycerides. Cholesterol esterase, which causes hydrolysis of cholesterol esters, and phospholipase, which splits fatty acids from phospholipids. Bicarbonate ions and water are secreted by epithelial cells of the ductales and ducts. Bicarbonate ion in the pancreatic juice serves to neutralize acid emptied into the duodenum from the stomach. Here you see the secretion of isoosmotic sodium bicarbonate solution by the pancreatic ductules and ducts. This is the lumen, and this is the pancreatic cell, and then the extracellular capillary in the extracellular area. What we see here, carbon dioxide and water will be combined to form carbonic acid in the presence of carbonic anhydrase enzyme, as you see. Then it dissociates into hydrogen and bicarbonate ions. When hydrogen is exchanged with sodium, sodium is secreted into the lumen with bicarbonate so that sodium bicarbonate or bicarbonate rich secretion or basic or alkalic secretion of pancreas is formed in this way. This alkalic secretion is important to neutralize the acidic chyme arriving in the duodenum. Here you see the sodium bicarbonate, water, and enzyme secretion by the pancreas caused by the presence of acid fat or peptone solutions in the duodenum. If there is excess hydrochloric acid, as you see, then bicarbonate-rich secretion occurs. When there are fats, then enzyme and bicarbonate are equal almost. But when there are proteins, then as you can see, the enzymes in the secretion of pancreas uh, are increased in the secretion, whereas bicarbonate amount decreases. Pancreatic secretion is stimulated by acetylcholine, cholecystokinin, and secretin. Acetylcholine or choline, which is released from nerve endings, mainly stimulates secretion of digestive enzymes. Cholecystokinin, which is secreted mainly by the duodenal and jejunnal mucosae, mainly stimulates secretion of digestive enzymes, and secretin, which is secreted by the duodenal and jejunnal mucosae when highly acidic food enters the small intestine, mainly stimulates secretion of sodium bicarbonate. Pancreatic secretion occurs in three phases. First, cephalic phase. Nervous signals that cause gastric secretion also cause acetylcholine release by vagal nerve endings in the pancreas. This accounts for about 20% of the pancreatic enzymes after a meal. And the second, gastric phase. In the gastric phase, the nervous stimulation of enzyme secretion continues, accounting for another 5 to 10% of the enzymes secreted after a meal. And the third, intestinal phase. After chyme enters the small intestine, pancreatic secretion becomes copious, mainly in response to the hormone secretion. In addition, cholecystokinin causes still much more increase in the secretion of enzymes. So regulation of pancreatic secretion, you can see here, acid from the stomach arrives in the duodenum, so it releases secretin from the wall of the duodenum. Fats and amino acid cause the release of cholecystokinin. So, uh, secretin and cholecystokinin are absorbed into the bloodstream. So, they affect secretion of pancreas. Uh, Secretin causes copious secretion of pancreatic fluid and bicarbonate. Cholecystokinin causes secretion of enzymes. And vagal stimulation releases of enzymes into the acini of the pancreas. So when chyme arrives, chyme is acidic, and then bicarbonate-rich secretion from the pancreas neutralizes it, so that preventing the ulcer formation here. It has enzymes, a lot of enzymes for three types of organic chemicals: carbohydrates, fats, and proteins. Secretin stimulates secretion of bicarbonate, which neutralizes acidic chyme. When acid chyme enters the duodenum from the stomach, the hydrochloric acid causes the release of prosecretin and activation to secretin, which is subsequently absorbed into the blood. Secretin, in turn, causes the pancreas to secrete large quantities of fluid that contain a high concentration of bicarbonate ion. Cholecystokinin stimulates enzyme secretion by the pancreas. The presence of food in the upper small intestine also causes cholecystokinin to be released from cells called eye cells in the mucosa of the duodenum, jejunum, and upper ileum. This effect results in particular from the presence of proteases and peptones, which are products of partial protein digestion and of long-chain fatty acids. Hydrochloric acid from the stomach juices also causes cholecystokinin released in smaller quantities. Secretion of bile by the liver. Functions of the biliary tree. Bile is important for first, fat digestion and absorption, and the second, waste product removal from the blood. So first, fat digestion and absorption. Bile salts help emulsify the large fat particles into minute smaller particles that can be attacked by the lipase enzyme secreted in the pancreatic juice. They also aid in the transport and absorption of the digested fat end products to and through intestinal mucosal membrane. Waste product removal. Waste product removal. Bile serves as a means for excretion of several important waste products from the blood, especially bilirubin, an end product of hemoglobin destruction, and excess cholesterol synthesized by the liver cells. Bile is secreted in two stages by the liver. The initial portion, which is secreted by liver hepatocytes, contains large amounts of bile acids, cholesterols, and other organic constituents. It is secreted into the minute bile canaliculi that lie between hepatic cells in the hepatic plates. A watery solution of sodium and bicarbonate ions is added to the bile as it flows through the bile ducts. This second secretion is stimulated by secretin, causing increased quantities of bicarbonate ions that supplement pancreatic secretions for neutralizing gastric acid. So you see here liver secretion and gallbladder emptying. So vagal stimulation causes weak contraction of the bladder, uh, but cholecystokinin released from the duodenum and in the presence of fat, uh, it contracts the gallbladder while relaxes the Otis sphincter here. So secretin via bloodstream stimulates liver ductal secretion. Bile acids via blood stimulate parenchymal secretion. And bile is stored in gallbladder and then concentrated 15 times. So, bile is concentrated in gallbladder. Active transport of sodium through the gallbladder epithelium is followed by secondary absorption of chloride ions, water, and most other soluble constituents. Bile is normally concentrated about fivefold in this way. So, composition of bile, you can see here water, bile salts, bilirubin, cholesterol, fatty acid, lysine, ions. Uh, I made some of them bold, as you can see how concentrated they are. So bile salts, bilirubin, lecithin, they are concentrated too much. Cholecystokinin stimulates contraction of the gallbladder. Fatty foods that enter the duodenum cause cholecystokinin to be released from the local eye cells. Cholecystokinin causes rhythmical contractions of the gallbladder and simultaneous relaxation of the sphincter of Oddi, which guards the exit of the common bile duct into the duodenum. In this figure, you see the formation of gallstones. Since bile is concentrated in the gallbladder, sometimes stones are formed there, and then these stones can enter the choledoct and then can block the choledoct. In this way, can prevent the transfer of bile into the duodenum so that the bilirubin concentration in blood will be higher so that we will see icterus or jaundice in these patients. So the reasons of the formation of gallstones: too much absorption of water from the bile, too much absorption of bile acids from bile, too much cholesterol in the bile, and inflammation. Okay. Secretions of the small intestine. Brunner's glands secrete alkaline mucus in the small intestine. Secretion of mucus is stimulated by the following. First, tactile stimuli or irritating stimuli of the overlying mucosa, vagal stimulation, which causes secretion concurrently with an increase in stomach secretion, and gastrointestinal hormones, especially secretin. Mucus protects the duodenal wall from digestion by gastric juice. Brunner's glands respond rapidly and intensely to irritating stimuli. In addition, secretin-stimulated secretion by the glands contains a large excess of bicarbonate ions, which add to the bicarbonate ions from the pancreatic secretion and liver bile in neutralizing acid that enters the duodenum. Intestinal digestive juices are secreted by the crypts of Lieberkühn. The crypts of Lieberkühn lie between the intestinal villi, and the intestinal surfaces of both crypts and villi are covered by an epithelium composed of two cell types. Goblet cells secrete mucus, which provides its usual functions of lubrication and protection of the intestinal mucosa, and enterocytes secrete large quantities of water and electrolytes in the crypts. They also reabsorb the water and electrolytes along with the end products of the digestion over the surfaces of the villi. You can see here a crypt of Lieberkühn found in all parts of the small intestine between the villi, which secretes almost pure extracellular fluid. Secretions of the large intestine. Most of the secretion in the large intestine is mucus. The mucus protects the large intestinal wall against excoriation, provides the adherent medium for fecal matter, protects the intestinal wall from bacterial activity, and provides a barrier to keep acids from attacking the intestinal wall. So, uh, in this presentation, we tried to cover the secretions of the gastrointestinal system. Thank you for listening.